non-monotonic dose-response curves. Well, they are complex and they are useful to organisms. They have been selected through evolution for some useful purpose. They are challenging for regulators, but anyway, from a biological point of view, they are very important and we need to understand them. Where is the clicker? Oh, here. Okay. So life will be absolutely simple if we had to deal with this type of dose-response curve, because as many regulators know and use, then if you just use a humongous dose, like here, you can have the effect, the only effect of such a phenomenon. And these curves don't change slope. That is precisely, I mean, you have an effect here, you can choose this dose, and then you know most of what you need to know. But biology is not so simple. As the great biologist Francois Jacob once said, Mother Nature is not a human engineer, it's just a tinkerer, and will make a cart out of a table. And evolution is what actually does, takes one thing and transforms it in another. And because of that, we have to deal with much more complex things that work very well. That is how we are alive, intelligent, etc. Sometimes intelligent, I should say. Anyway, so this is a very old story for us endocrinologists. And this is one of the papers, I mean, I was already working on that, when Priscilla Danis published this paper with a cell line that was developed by my partner, Carlos Sonnenschein, where she was showing this beautiful non-monotonic dose response of estradiol regarding cell proliferation. In these very same cells, she was measuring the secretion of prolactin. And so you see, this is the activity of just one gene that responds to estrogens with a monotonic dose response. The same cells, regarding proliferation, are induced to proliferate at very low doses, 10 to the minus 11 molar is the peak, and then as you increase the dose, they proliferate less. So this is a safe mechanism, as Jack Gorski said at that point. He was a very prominent estrogen receptor researcher, that nature has put a brake there. So estrogens and progesterone that are proliferative in sex accessory organs produce this big event of proliferation during puberty. But then you have to regulate that. And the doses of estrogens and androgens that occur at that point tend to block this proliferative effect, but they don't block the induction of useful proteins. So you have the two things. It's not a toxic effect, it's a regulatory effect. So I will give you an organismal perspective of the usefulness of this non-monotonicity. As I said before, the same hormone is able to modulate its own effect. And you can see that because the proliferative and inhibitory phases occur through different mechanisms. I am going to show that later. Androgens, for example, increase prostate size by inducing cell proliferation, but at puberty, the high levels of testosterone are established, and they are constant. And so when the adult size is reached, androgens inhibit cell proliferation. That really blocks men from being a prostate with legs. But so it's a very important thing, I would say. Animal strains that develop pituitary cancer lack the estrogen-induced proliferative shutoff that was described by Priscilla Dennis, and I showed you 2 minutes ago. So if you give estrogens to these animals, in a few months they develop a pituitary tumor. But if they have conserved that mechanism, they never do. Additionally, chronic administration of high estrogen level inhibit mammary gland development, and that is cancer development. And that was discovered in the mid-60s, that you take the animal, you give a carcinogen, and the animal has her ovaries, it will have a certain latency period that is short. If you put a pellet of estradiol, that latency period is much, much longer. So, and even those estrogens could block the development or make the tumors regress. And before tamoxifen was invented, that was the choice treatment for breast cancer. And here we see non-monotonic dose-response curves that are, as I said, are remarkably common. So here, this is from our lab. And this is, I'll explain the details later because I'm going to use the same slide. But here is at tissue level of organization. Those are terminal buds that are some particular structure of the mammary gland. And the more estrogens you put, well, the more of them you get, up to a point where it really inhibits further development of the mammary gland. And this is in vivo, obviously, and is the result of multiple interactions at different levels of organization. That will be explained later. And here, a phenomenon that we describe in cell culture, whereby prostate cells, epithelial prostate cells, proliferate maximally at a dose of synthetic androgen, R1881, that is about 10 to the minus 10 molar, and then that gets inhibited. If you use the dihydrotestosterone, you obtain the same thing, but at higher dose. And what you see there is that you have this effect on cell proliferation. However, the synthesis of prostate-specific antigen is not inhibited at any dose. So, what are the mechanisms underlying this phenomena? Well, the first one that was discovered is receptor downregulation, and I'm going to show you that. Then others that have been described are cell and tissue-specific receptors and cofactors that are different from one way of the phenomenon and the opposite one. Receptor selectivity, receptor downregulation, endocrine negative feedback loops, and tissue interactions. So we have a lot to cover. The idea here is quite simple. So you have a membrane receptor, that is this thing that looks like a Y, and then this rounded thing that is the hormone. So when binding occurs, the complex hormone receptor is internalized, and so you have this early endocytic event. Those endocytic vesicles fuse with lysosomes, and then the receptor is degraded. And we see that here, that binding of the hormone to the receptor is maximal during the few minutes after you add the ligand. But then you see a decrease, and that is due to the fact that the receptor is being chopped up in the lysosomes. So that blocks the initial effect of the hormone. And here you see the result. In this case, is my favorite hormone, estradiol. And so you see that here you have concentration of estrogen receptor, and here you have the dose, and you see that at the beginning you have all this binding, and then it goes down because the protein has been degraded. Okay, that is very simple. Now you have other effects. This is also from my lab, and this is the effect of certain phytoestrogens. And it's interesting if you look at this curve. So this is a phytoestrogen alone. So you have this very sharp increase and then decrease on cell number. If you add estrogen, estradiol, you see that because it's more potent, that you have a proliferative effect earlier. But you see that high dose of daidzein inhibits the effect of estrogen. So this effect is mediated through the estrogen receptor. This we do not know, but the fact that cannot be blocked by a competitive anti-estrogen indicates that occurs through another mechanism. People say that it's through the inhibition. It hasn't been really demonstrated very clearly, but is claimed to be an effect through inhibition of tyrosine kinases. Be it as it may, it's two separate things. And you can see here how this is the effect of estradiol, a humongous increase of the cells in S phase. If you add a humongous amount of daidzein, one of those phytoestrogens, you get it back to control. And so again, it inhibits estrogen action. So there you have another non-monotonic dose-response curve. And here we get back to the prostate cell lines that I showed you before. So we see this effect. So what we did, and this is quite complex, we did, at that time, we didn't have all the omics that we have today, so we are in the late 80s. So what we did is a double subtracted library to know what was the difference between the gene expression at this point and at this point. And so we obtained a protein that is a transcription factor among the candidates that we call aprin, that is a transcription factor that controls chromatin packing. So at low androgen doses here, we see all these cells with their nuclei labeled with BDR, that is, they are proliferating, incorporating nucleotides into DNA. And here you see that that is inhibited, okay? And the inhibitor is aprin. We have put aprin under control of an antibiotic, tetracycline, and so we can produce this arrest of cell proliferation without androgen, for example. So we demonstrated that the second part of the curve occurs through the induction of aprin. And this is a paper, well, the senior author is here, Maricel Maffini, where we tried to do the same thing in the prostate, the rat prostate. And here you see, in the adult male, you have all this aprin being expressed. That means that the cells are not proliferating. You can also incorporate BRDU, and you won't see any or very few cells labeled. If you castrate the animal, of course, all this regresses, and there is no aprin. If you put androgens, the first response is an effect on the differentiation of this gland, and also proliferation. You won't see it here, but in the next. And then if you prolong the exposure to androgens, you would see, again, the expression of this inhibitor of cell proliferation. In the next, what we see is that when the prostate is growing, you see mostly BRDU-labeled nuclei. But when aprin is induced, you see very few of these cells proliferating. So we had enough of native hormones. This is an old phenomenon in biology. Let's go to endocrine disruptors. And if you look at BPA, very low dose of BPA normally is claimed to act through membrane estrogen receptors, and sometimes the evidence is very strong, like in the case of Nadal and Watson's. And then intermediate dose, you have most effects occurring to nuclear estrogen receptors. I mean, when we say that, here, in particularly Angel Nadal's experiments, is the same receptors as we call nuclear, but they are located extranuclearly. High doses act through thyroid receptor, and through androgen receptor. So we have different effects according to the dose because they activate different systems of receptors. In other cases, what is being affected by this non-monotonicity is cell-cell interaction. So here we are in the uterus, and if we have only epithelial cells, there is not much effect by estradiol. But if you put epithelial cells and uterine fibroblasts of the lamina propria, you obtain an effect. And this effect is induction of progesterone receptors. Progesterone activation blocks the effect of estrogens. So you will have a proliferative phase, and then when progesterone receptor is induced, you will have an inhibition of the proliferative effect. And now we are going to talk about more complex phenomena that occur at the tissue level, such as the maturation of the mammary gland. So here you have a structure that is called terminal embud. This is an invading organ, and here we see it in the real world histology. Here we see a kind of simplified version. What is happening there is that there is cell proliferation and invasion. You see the cell proliferation. Here the cells are labeled with bromodioxyuridine, and the lumen is formed because there is cell death. So you have a combination of two phenomena, cell proliferation and cell death. And the effect of estrogen here is to produce a non-monophasic dose response, whereby we have here, these little things are the terminal embuds. Look at this. So estrogens do two things. One, here is the front of invasion. You see that in the control there is not much invasion past this structure that is a lymph node. Here you have all this invasion, and we are measuring it here, and you see that is non-monotonic, it's biphasic. If you count the terminal embuds, you also have a non-monotonic dose response. And if you count the area of those structures, you also have that. Okay. If, on the contrary, you look at the uterus of the same animals, before I get there, if you look at the gene expression, induction of progesterone receptor, induction of Wnt4 and MSX2, all of them are monophasic. Okay? Gene by gene is monophasic. Tissue-wise is biphasic. If we look at the uterus, you see that the effect at tissue level is also monophasic. This is the uterus of the same animals that have a biphasic response in the mammary gland. The same mechanisms that we have seen in the normal mammary gland occur in mammary cancer. And here you see that, oh my God, this is too sensitive, or I'm not so coordinated, one or the other, or both. Okay, so here you have number of tumors per animal, and you see that it's biphasic. Tumor volume, same thing. Then if we look at tumor volume and proliferation index, we see the same thing. Here you have, this is just the first phase, that is the increase on cell proliferation. And you see that it peaks at 25 micrograms per kilo. If you look at the rate of apoptosis, it maximizes at 2,500. When you add up one and the other, what do you get? A non-monotonic dose response. Okay? As seen here. So it's the addition of this ascendant branch and this descendant one. And now I will talk about something else. That is the Clarity study that many of you may have heard. This is a brainchild of Jerry Heindel that is here. And the idea was to put a group of academic researchers together with a guideline-compliant study. So I was one of these principal investigators, and I hooked to this project, to the main project, by asking questions about the dose-response curve of BPA on the mammary gland. So my objective was to determine the effects in the developing mammary gland and evaluate those later on when the animal is an adult at 90 days of age and 6 months of age. The methods we use is to expose to, well, actually the FDA did the exposure, and we compare negative control, ethinyl estradiol, that is a positive control, and then different 5 doses, actually, of BPA. And so we looked at whole mounts and histological specimens. What we observed, and this is unpublished, by the way, is being reviewed right now, is that at day 21, that is prepubertal, we found a clear non-monotonic dose response with a breaking point between 25 and 250. So it's not a biphasic, it's just, I'll show you the shape in a minute. It has a breaking point. This breaking point was also observed at PND 90 and at 6 months. And the BPA response was different from ethinyl estradiol for many features, and for others it was the same. Now, the quantitative unsupervised analysis that we use define 91 measurements. So that is very, very, very heavy data. It's very, very solid because we have 91 endpoints different. And most of them had a very striking non-monotonic dose-response curve. Lower doses resulted in larger effects, which is consistent with what we knew already. And moreover, in the core study run by the FDA, tumors were significant at the lowest dose, that was 2.5 micrograms per kilo, which coincides with this. So, in this study, this is about the dose-response curve that we observe for most of these 91 parameters. And here you have what is that we are measuring here. We call it the standard deviation of the width of the branch, and that represents the appearance of what is called buds. You see here, because that is read by our method as a widening of the duct. Because this is totally blind. It's done by a machine. We have nothing to do with this. OK, so here you see low value. It looks like this. It's this point. High value, it looks like this. OK? So I think it's pretty clear. And you see it over and over and over in 91 different points. So I'm not going to show all that because I have only 5 minutes. OK? The number of observed quantities in those 91 were also analyzed this way. What was the difference of effect between 2 consecutive doses? And you see that for pretty much most of them, the big difference appeared in between 25 and 250. You see 17 here, 0, 0, 57, 5, 6, etc., etc. So it's a very clear phenomenon. When you look at the effect of bisphenol A and ethinyl estradiol, you see that sometimes bisphenol A has a lower effect than control, and ethinyl more than control. So you see this pattern. Sometimes the pattern is that control is more than, that BPA is more than control, and then you can have another patterns, so that is not exactly the same. Okay? So I would like to conclude that many of the same mechanisms that operate in vitro are observed in vivo, as I show you for the prostate. Tissues are more complex, and you will have very different effects because it depends on the 3D structure of the tissue, the different cell types that interact, etc. And now I would like to ask you, do we need to know the underlying mechanisms to accept the existence of non-monotonic dose response? And I'll give you a very clear example why we shouldn't put the cart before the horse. Since time immemorial, we practice castration of farm animals, which we knew that they didn't reproduce, but we have not the slightest idea about what. 500 years ago, we didn't know, or 5,000. So we do not need to know mechanisms in order to accept this, the existence of biological phenomena. Nonetheless, we do have mechanisms here, okay? And they occur at all levels of biological organization. They occur with natural hormones and endocrine disruptors. The mechanisms that I show you are very well understood. And the existence of a phenomenon is not defined by the mechanism producing it. It's a phenomenon that exists at a higher level of organization. And it has importance from the organismal and evolutionary aspects of biology. And with that, I thank you for your attention. And I hope that I have brought to your mind the powerful tool that is this non-monotonicity from a viewpoint of an organismal biologist. This is normal in endocrinology and should be considered when you look at endocrine disruptors. Thank you.